Hello friends, and welcome to our third video in this series. Let's learn cells. In this tutorial, we will learn how to search for a sequence if you do not know its accession number, how to apply filters, perform advanced search, and fetch data from gene features table, and finally, how to get FASTA format sequence. We will go to NCBI homepage and choose Nucleotide Databases from the menu. We can perform a general search or advanced search here for any gene. First, we will type a general search for a gene named retinoblastoma or RB from humans or Homo sapiens. RB is a tumor suppressor gene and its function is lost in many cancers due to mutations in this gene. Our search gives 7,072 entries. Please remember that our results may vary as databases get updated on a daily basis. Therefore, there may be additions or deletions in these entries. Now, 7072 is a huge number of results and we need to make our search more specific. Our first filter is for Homo sapiens as organism on right hand side, which reduces the entries to 1115 by removing the RB gene entries from other organisms. Next, we can apply a filter for genomic DNA or RNA sequences from left-hand side filters, which further narrows down the entries to 250. If we want only INSDC, which is GenBank sequences, we can apply this filter also to further narrow it down. Now the results show 212 entries, which is still large to choose one specific result, even if we ignore any chromosomal assemblies, predicted or partial sequences. But still there are many and it's confusing. Please remember that in our example, we do not know any specific accession number. The only information we have is name of the gene and the organism we wish to retrieve data for. We will now try advanced search option. You will notice advanced written just below the text box. Click on that. Nucleotide Advanced Search Builder initially shows two fields joined by a Boolean operator field. Each builder further has many fields to search for, like gene name, organisms, keywords, author, etc. You can choose an appropriate field based on the information you have and fill in the details like gene name, RB, which is short for retinoblastoma gene, and a second field can be organism. Advanced option also allows us to use three choices of combining the fields by Boolean operators and, or, and not. Here we have filled all the information we have, like gene name as RB, Boolean operator and organism as Homo sapiens. It's good to go by hitting the search button now. Alas, and here we get only 7 results. Far easier to search which one is the most appropriate. It's so much better. Now let's choose the one which is most appropriate. Item numbers 2, 6 and 7 are of similar size and all are mRNA molecules. We can either tick the box we wish to work with or click on the link directly. By clicking on the link for the second item, I got this description which is called Annotations. So here are the annotations for the RB gene from Homo sapiens. Let's have a look at the annotations. The sequence has been taken from the ref sequence database of the NCBI nucleotide and its accession number is NM_000321.2. It's an mRNA and 4,772 bases in size. By definition, it is a Homo sapiens RB transcriptional co-repressor. Let's scroll down and there are quite a few references associated with this sequence. We will now see features of this gene as indicated here. Clicking on gene will highlight the whole sequence in GenBank format. Other gene features can also be checked from a box on bottom left corner like CDS, which is the complete sequence, and exons, etc. Let's see how many exons, which are the protein coding regions of the gene, are present in RBG. Choosing exon will highlight the first exon and show first of the 27. Hence, there are a total of 27 exons, and we are seeing the sequence of the first exon of RBG from Homo sapiens. You can navigate to other exons from here. Similarly, choosing regulatory feature options shows a highlighted poly A signal sequence, which is a signal to add poly A sequence to the RB mRNA by specific enzymes. Choosing poly A site will highlight the base where poly A tail will be added. Here base C or cytosine is highlighted. Finally, we want the sequence to be used for various purposes like BLAST or Clustal W software analysis or any other software analysis. The given sequence here is in GenBank format, which has spaces and numbers. Most softwares or tools will not accept this format, hence we need to get the sequence in FASTA format. This is also indicated at the beginning of the annotations. FASTA is an abbreviation for fast algorithm. FASTA formats are used for nucleotide as well as for the protein sequences using single letter codes. By clicking on this link, the FASTA format sequence will be available for further use. Here the first line with accession number, etc. is called a descriptor, and the sequence begins from the second line. You can copy and paste the FASTA sequence in softwares, but always include the descriptor line in your analysis. Moreover, we can run a BLAST analysis using a link Run BLAST on right hand side for the given sequence. In our next video, we will learn more about what is BLAST, its types, and how to run and analyze BLAST end results. Hopefully you found this information useful. Hope you are enjoying these tutorials. Please like and subscribe to my channel and keep me going for the next series. Thank you so much and see you soon for the next video.